Hi everyone. Today we're of the animal cell. We mentioned yesterday that the animal cell is not the only kind of cell. There are plant cells and there are prokaryotic cells. We even have things like fungal cell will have different changes to it. But the animal cell is a pretty famous and pretty all-encompassing kind of cell. And so we're going to start our look at cells by looking at the animal cell. This is a basic animal cell here, and as we get through the chapter, we're going to actually come back to this at the end. But this is something kind of similar to the project I'm going to have you guys start on Wednesday, where you're going to start kind of drawing a cell. It includes all of the important organelles like the Golgi and the ribosomes, the rough ER and the smooth ER, which is over here, lysosomes, the cell membrane. And in this particular drawing, it has equated these things to being like in a factory. The assembly line, the workbenches, the distribution center. And so we're not going to be making an analogy like that for your assignment, but I am going to want you to be able to describe the role and the purpose of each of the different organelles inside of the animal cell. We are going to break our eukaryotic cell into these categories, which is how your book breaks down the components of the cell. We're going to start by looking at the nucleus, and then we'll look at the organelles that build, that are responsible for building. There are organelles that are responsible for energy. There are organelles that clean and support, and then there are the boundaries. And what you'll notice is that two of these structures have this little plant next to them, which means that these are exclusive to plant cells. So we're not going to really be looking at them in the animal cell, but we'll come back to them later, and they go into these categories. So we'll come back to them when we look at plant cells. So we're just going to jump right into it. We're going to get started looking at the nucleus. The nucleus is what holds and protects your genetic information. The nucleus has a membrane that goes around the outside as a protective layer. Anytime you hear the term membrane, you should be thinking protection. Membranes protect and maintain a boundary. So if you have a membrane, you could think of that as being like the walls to your house. But then the nuclear envelope, which is out here, or the nuclear membrane, might be like the walls to your room. It's an extra layer of protection. So if you are out in your house, you have one level of privacy, but if you close the doors to your room, you have an additional level of privacy. And that's really what the nuclear envelope is for. It's going to protect the DNA to make sure, especially sure, that the DNA doesn't get damaged, because the DNA holds all of your genetic information. You really don't want the DNA to get damaged. Inside of the nucleus, there is this area which is called the nucleolus, and it is the darkest staining portion of the cell. The nucleolus is where ribosomes get assembled. So this is where we start to put together the ribosomes, and we'll be talking about the ribosomes later. They're going to be on the rough ER, and they're going to be all throughout the cell. But we make ribosomes in the nucleolus, and the nucleolus is inside of the nuclear envelope. The other thing that's inside of the nucleus is, of course, the DNA. And the DNA can be in two different forms. It can exist as chromatin, or it can exist as chromosomes. DNA packs into chromosomes when we're getting ready to move, when the cell is getting ready to divide. Dividing your DNA is one of those just don't make a mistake when you're doing it kind of situations. You have to keep your DNA safe and protected. So I want you to think about going on a trip where you're going to the airport, and think about showing up to that trip with your clothes for your trip just in your arms, like if you're carrying them to the laundry. I don't know if you guys carry your clothes in a big like bear hug to the laundry. That's how I usually carry them as I go around the house collecting them. And you know, you kind of like keep adding clothes to the top when you've got this kind of big section of clothes, and you carry them. Now they always kind of fall, right? Like, oh, I lost a sock, and so you reach down, you grab the sock, and you like add it to the top, and you keep walking. Think about going to the airport with your clothes like that. and you get to the airport and they're like, okay, well, we're going to check some bags. And you're like, oh yeah, sure. And you just, boom, dump all the laundry onto the conveyor belt. Can you just think about the looks that they would give you? Oh my gosh, it would be crazy. We don't travel like that. Why not? Well, they would lose your luggage, right? What are the chances that your clothes make it from LAX to Hawaii? Let's go to Hawaii. If your clothes make it from LAX to Hawaii without being put in a bag, they just get thrown onto the airplane and you just hope that they make it there. No, that is not a good plan. If we're going to travel, we pack all of our clothes really nice and tight. You roll up your shirt, maybe you roll up your whole outfit into one bundle and you keep them together so you've got whole outfits you can pull apart. It's like such an amazing packing trick. But you're going to pack up and you organize your clothes. And first you put them out on your bed and then you make sure you have everything that you need, and then you take it really nicely and you put it in your bag and you zip that bag closed, and you're sure that that bag is going to make it from LAX to Hawaii. Then you get there. When you arrive to your destination, do you keep all of your clothes packed so tightly and neat and wound up? Oh, heck no. I could be just going overnight, right? And I'm immediately going to open up my bag. Everything's coming out. I got to look at everything. I've got to rematch all of my outfits. I've got to think about what I'm going to wear. All the clothes go exploding, right? Now it's time to leave again. You pack all the clothes nice and tight, nice and neat into your bag, and then you travel home. Well, DNA is no different. We need to use the DNA on a daily basis. And when we need to be using that DNA, we leave it as chromatin. And as chromatin, it's this kind of stringy stuff that we see in the nucleus. Only when the cell is packing for a move, for a cell division, does it pack that chromatin down into chromosomes. And we know that we have 23 chromosomes. Maybe you didn't know that. Now you do. Humans have 23 sets of chromosomes, or 46 total chromosomes. That's why they call it 23andMe, because it's telling you about your DNA. We have 23 sets of chromosomes, and we pack those 23 bags and we line them up 2 by 2 to make sure everything is in its place before we do our cell division, to make sure that each cell is an exact replica and no socks get left behind, because a sock getting left behind, as it were in DNA, is going to be the starting of mutations that are going to end up leading to things like cancer, and we don't want to see that. So we don't want any deletions or additions or transpositions of our DNA. We want to keep our DNA exactly as it was. And so that packing of the chromatin into chromosomes is a really important step. So the DNA in our nucleus can be either in the form of chromatin or in the form of chromosomes, just depending on the scenario, the moment of what's happening inside of the cell. Beyond the nucleus, we are going to make proteins. And so the organelles that build are the organelles involved with making, packaging, and shipping proteins. Ribosomes, rough and smooth endoplasmic reticulum, or ER, and the Golgi are all responsible for proteins, making, shipping, packaging proteins. Ribosomes do the hard work. So a ribosome is just a thing. It's a small little portion of the cell. And the ribosome is where protein synthesis happens. So we know that the instructions for making a protein are stored in the DNA, but the machinery, the organelle that actually makes the protein, the physical doer of making the protein, is a ribosome. So this green sphere is the ribosome. These little colored things here are turning ultimately what was our DNA, our genetic information, into a protein. But the place where that happens, the site where that happens, is the ribosome. And so the ribosomes can either be free, all spread throughout the cell like little bits of sand, or they can be attached to the endoplasmic reticulum, making it the rough ER. So these little purple dots right here are the same thing as this big green sphere, and everywhere you see these little purple dots all throughout the cell, whether attached to the ER or floating by themselves, everywhere you see those little purple dots, proteins are being made. So those are the places where proteins are manufactured. Once manufactured, those proteins are destined to circulate throughout the cell. So the ER, or the endoplasmic reticulum, that's why we call it ER, that's a mouthful. The endoplasmic reticulum is like the circulatory system of the cell. You can see that the endoplasmic reticulum is continuous with the nuclear envelope. So in here we have the DNA, and then we have this nuclear envelope going around the outside, or a nuclear membrane, and then continuous with that, we see the rough ER, and then continuous with the rough ER, we see the smooth ER. The rough ER is responsible for proteins because there are ribosomes on it. That's what makes it rough. That's how we know that the rough ER is involved with proteins, because we just said ribosomes and proteins go together. So if the rough ER has ribosomes, then the rough ER makes proteins. The smooth ER, instead of making proteins, makes lipids and detoxifies. It cleans a lot of what happens in your cells. So your liver, which is an organ in your body that's responsible for detoxifying, the cells of your liver have lots of smooth ER, because the smooth ER is the organelle that is responsible for detoxifying. Our bodies are incredible, but when you look at the tissue in the liver and you look in liver cells, you will see an abundance of the organelle that does the actual detoxifying of that smooth ER in there. But the smooth ER also does some lipid modifications. It helps with making cholesterol and some other lipids for our body as well. So smooth ER is all for our lipids. Rough ER manufactures and distributes proteins. Now remember, the actual site of the protein synthesis is the ribosome, but the ribosome could be attached to the rough ER. Once our proteins are made, they're going to be moved, transported from the rough ER to the Golgi body. The Golgi body is sort of like the post office of the cell. It receives all of the proteins that were made in the rough ER, and then it reorganizes them. It's collating them. It's going through and saying, okay, I need 5 of these proteins and 2 of these proteins and 6 of these proteins, and then we're going to put these all together into a bundle, and this is going to do a specific job, and I'm going to go send it out to do that job. One of the jobs that it could go do is secrete that information from the cell out into the intermembrane space. So it could send that information out. Maybe it's a cell signal, maybe that's a hormone, maybe it's some sort of antibody, like the cell noticed a problem, made some protein antibodies. Those antibodies got modified, and now they're being sent out to go fight disease. So the Golgi is going to modify, package, and ship proteins out from the rough ER, receives from the rough ER, sends them out either as secretory vesicles to be secreted into the intermembrane space, or we're going to talk about another destination they could have if they stay as a lysosome. One of the last things we're going to talk about today is this big picture break. What we're seeing here is that our rough ER makes proteins. These proteins then get transported to the Golgi in something called a transport vesicle. And then the transport vesicles then leave the cell and get secreted into the intermembrane space. The information to make the proteins is here in the nucleus. That information comes out into the rough ER. Rough ER makes proteins, sends the proteins to the Golgi. Golgi modifies and packages them, and then sends them out to be secretions. The other option is that we could make a package that we don't send out. Instead, we make a package that we keep, and that package is full of defensive, digestive enzymes. And so if a foreign invader comes into the cell, we can send these lysosomes, which are little packages of digestive enzymes, we can send the lysosomes to go attack and kill and break down that foreign invader. So sometimes the Golgi makes packages that are going to be shipped out, and other times it makes packages that we're going to keep for the purpose of the health of the cell. So this is looking at this process. So you can see that the Golgi make these little transport vesicles full of protein that leave and travel to the Golgi. When they reach the Golgi, they move through the Golgi apparatus, the Golgi complex, the Golgi body, whatever you want to call it, and then they're shipped out in a secretory vesicle. And the secretory vesicle then merges with the membrane and excretes or secretes the proteins that had been packaged into the secretory vesicle. Okay, so we have looked at the nucleus, we have looked at the organelles that build proteins, the ribosomes where they're made, the rough ER where they're moved, and the Golgi where they're modified and shipped. The last thing I want to look at today are the organelles for energy, and this is the first time we come to one of these plant ones. So we're not going to talk about the chloroplast just yet. We're going to wait till we get to plants. So the only organelle for energy in the animal cell is the mitochondria. So right now we're going to take a little look at what we see in the mitochondria. A mitochondria is the powerhouse of the cell. It converts food, chemical energy from food, into ATP, which is the energy that cells can use. So when you eat dinner, that turns into energy. The mitochondria is the organelle in all of your cells, in all of your body, that turns that food energy into body energy, ATP, that we can go do something. Cells can have a few mitochondria or many mitochondria, depending on their energy needs. So if you're looking in a tissue of your cell, and that tissue does a lot of energetic work, that tissue will have many mitochondria. Mitochondria are double membrane bound organelles. They have two membranes. So we're going to look inside of this mitochondria. It has an outer membrane that is dark brown, and it has an inner membrane that is more beige. This inner membrane is called the cristae membrane, and it has a bunch of folds to have more surface area. That's a problem for another day. That's something we'll talk about later. But what's really interesting about the mitochondria is something called endosymbiotic theory. It's a big word. Endo, in. Symbiotic, together. So the idea is that the mitochondria was probably once a free living cell that got engulfed, brought in to the animal cell, and instead of being eaten, was saved and kept as a mitochondrion. And so it's kind of like the domestication of a dog, where it used to be a wolf, but over time and domestication, it's become a part of our lives. And so we have this symbiotic relationship where you have the dog and you feed the dog. And in turn, the dog offers you affection and protection. I don't know if you guys have ever seen The Princess Bride, but how Wesley becomes the Dread Pirate Roberts is actually very similar to how I think the mitochondria became an organelle in the cell. When a mitochondria came into the cell for the first time, it should have been killed by those lysosomes that we talked about. Foreign invader, lysosomes bind, break apart the mitochondria. But instead, the mitochondria was especially good at making ATP through something called aerobic respiration. We'll come to that later. But it knew like a magic trick to make extra ATP. So when the cell brought it in, instead of eating it, it recognized the value of the mitochondria, that the cell was able to use the mitochondria to make all of this ATP, which allowed the cell to grow bigger, to live an easier life, all of these wonderful things. So the mitochondria stays inside of the cell. And the cell, in turn, gives the mitochondria a steady supply of energy, protects the mitochondria from the outside environment. I mean, the mitochondria was going to die. That's what happened when it wound up in the cell. The animal cell now protects it and provides it with energy. And in turn, the mitochondria makes huge amounts of ATP that the cell could never make on its own without it. And so the two have become so intertwined that if you take the mitochondria out, it can no longer survive. And if you remove the mitochondria from an animal cell, it will no longer survive. They're so dependent on each other that they have become one and the same. But when you look at the mitochondria, it has its own circular plasmid of DNA, just like a bacteria does. The mitochondria reproduces itself in response to the environment. So we said busy cells have many mitochondria. When a mitochondria is getting overtaxed and its job is getting too big, it replicates from within the cell. The instructions to make a mitochondria are not stored in your DNA. I find that fascinating. You inherit your very first mitochondria from the egg cell of your mother. And that mitochondria goes on to reproduce enough mitochondria for your whole entire body. That's amazing. Your mitochondria are so amazing. If you want to check for the lineage of a person through their matrilineal line, their mother's line, you can actually trace it because mitochondria DNA is almost exactly identical between offspring and their mothers. So your mom and her mom and her mom and her mom and her mom, you all have the same, very close to the same mitochondrial DNA in your bodies. All right, so when we come back on our next video, we're going to talk about the organelles that clean and support, as well as cell boundaries.